Hi, thank you to the organizers for inviting me to speak, and I'm happy to share a few quick highlights from our hot spring metatranscriptome. But if you want to follow up later, I will also be at the poster session tonight. So just a quick introduction to the hot springs. Hot spring phototrophic mats form in the outflow channels from hot springs as the water cools from the source. These are typically found 70 degrees and lower Celsius. And if you take a core sample, as you see in the middle here, you have a nice layered structured microbial community. And as we'll be talking about today, these are made up of over 70 different genera, but we can kind of classify them into really four main categories: two different types of phototrophs and two different types of heterotrophs, which we'll go into more detail. So in these hot spring mats, so everyone, we focus on, wow, they grow at such high temperature, but I want to focus on a different stressor today, in that over the day-night cycle, these mat organisms experience large environmental shifts. So during the day, as cyanobacteria do oxygenic photosynthesis, oxygen increases, pH also increases, and we also find very minimal hydrogen. But at night, the whole system switches to an anoxic environment. pH lowers due to fermentation, and we also find an increase in hydrogen concentrations. And while we know a lot about how the cyanobacteria are contributing and responding to this, we know a lot less about the other organisms in the mat. So we want to know more about how does the mat respond to the day-night cycle, who is active, who is responding when, and how. So to do this, we were granted a CSP from JGI to do a number of different omics samples, including 34 metagenomes on a variety of different mat samples, which we were able to use to assemble and bin into MAGS, metagenome assembled genomes. In parallel, on 60-degree samples, we conducted some metatranscriptomes taken every 2 hours over a 24-hour period. We mapped these RNA reads back to our MAGs, and that's the data I'm going to show you today. How are the active organisms by RNA? So first thing is, at this temperature, 60 degrees, we found only 8 genera actually were above 1% or more of our mapped reads. So there's really a small number of really active species. So first, we have our oxygenic phototroph, our cyanobacteria synechococcus. These are organisms that use light energy, are able to fix carbon, and also produce oxygen using water as their electron donor. There are also 5 genera from 3 phyla of different anoxygenic phototrophs. I'll always be referring to these guys in orange here. These guys use a variety of different carbon sources and a variety of different reductants to do their photosynthesis. And we also found 2 heterotrophs. We're predicting them to be aerobic heterotrophs. These are both from Armatidomonodota. You might know them as OP10. And they're from 2 different families of the Armatidomonodota. So that told us a little bit about who was there, who was active, but when are they responding? So taking just a very big picture approach of this, if we look at just the a subsample of the genes over the day-night cycle, so I'm showing you heat maps where at the most active point, each gene is going to be yellow. So we can see, just looking at our 8 organisms here, there are very different patterns in total gene expression. For example, our OTU12-like heterotroph has a ton of active genes at night. In contrast, Thermochlorobacter has most of its active genes during the day. And I want to highlight actually our 2 Armatidomonodota, in that they actually seem to have very different patterns in peak expression. In that OTU3-like seems to have a lot of genes active during the day, and OTU12-like seems to have a lot of genes active at night. We did some analysis of the MAGs, and we find that OTU3, we think, is using amino acids, TCA cycle intermediates as its main carbon sources, whereas OTU12-like seems to be able to use more sugars, polysaccharides, some of these amino acids as well. And in addition, it has a hydrogenase. So our hypothesis is that these carbon sources have different timing of availability in the mat. So that was a little bit about when they're responding, but we want to know more about the biological activity. So I'm going to very quickly walk you through a discovery method we use to try to understand this. So we used a approach called consensus module detection with weighted gene correlation analysis. So very brief summary, we had 3 actual metatranscriptome time series, and I'm going to show you 3 examples of 3 genes. So first, in each time series, you detect which genes are co-expressed, which genes have very similar expression patterns. For example, gene 1 and 2, I've highlighted them in red, are very similar. So that's each time series independently. Now we calculate the consensus modules. So we look at what co-expression groups are consistent across all 3 time series. In this case, in our example, gene 1 and 2 have consistent expression in all 3 time series. And finally, to summarize all these data, we use what is called the eigengene to summarize the relative expression pattern in each group. So I'm showing just 2 genes here. These groups have hundreds of genes in each group. And then from that eigengene pattern, we're able to summarize what time of day each gene is expressed at its maximum. And then finally, to try to bring back biology, we're using functional groups using the COG categories, which represent a variety of biological features, and using overrepresentation analysis to try to discover when these features are most active. So just to walk you through what you're going to see, here's an example of our 2 ARMAs. Each row is a co-expression module. We've been able to classify some of them based on the peak expression times, either as evening-night peaking, night-morning peaking, or day peaking. So you can see our OTU3's got a lot of genes in the morning, a lot in the ARMA 12 has a lot of genes at night. And then each column is a COG category. And if it was significantly overrepresented, it's now highlighted in black. So here's a ton of data, and it's a lot to look at. But I want to show you that every organism seems to have its own different patterns. However, we found some conservation. I've highlighted with stars here that this COG category J, involved in ribosome biogenesis and translation, has peak expression in the morning in 7 of our 8 genera, which is suggesting that everyone is turning on their biosynthesis at a similar time. One other quick vignette I want to show from this data was that looking at Chlorcidobacterium, one of our anoxygenic phototrophs, we noticed that this COG category O was significantly enriched in a night peaking module. This included genes chaperones such as GRO-EL, depicted here. And this was really surprising because more than 3/4 of its genes are day peaking. So we're like, why is it turning these genes on at night? So we wanted to look a little bit more into that and see if that's something that's happening across all the organisms. And so here's the GRO-EL, for example, plotted for all 8 of our main genera. So we can see Chlorcidobacterium has that peak at night. We actually see that with a few other species such as Rosilunea and Rosiflexus. And our hypothesis for the phototrophs, at least, that perhaps this is related to the peak assembly time of the photosystem. So here I plotted the expression of the photosystem reaction centers. But there's a lot of other things that chaperones could be involved in, so I'd be really happy to chat with anyone about their ideas on why we might be seeing temporal expression of chaperones. So that was just a very brief blitz of some of the things we've seen in our data set here. I want to say that we would really like to use some of these techniques that were presented earlier this morning to try to improve our understanding of the community. And then for the ones we are able to have in culture, we'd like to better understand their regulatory response and what's controlling the expression of these genes. So I'd like to thank my lab and our collaborators and our funding. And with that, I'd be happy to take questions.
Great talk, Amanda. You explained the hot springs really nicely. I'll do less of it tomorrow. I was curious, when you look at these interactions through the, through your WCN, GCN analysis, is Synechococcus driving the ecosystem? If you took Synechococcus out of the system, would it fall apart? is there a way that you can try to figure out what is the real role of, or what is the driver of these math systems?
Yeah, so that was a great question. The question was whether we could, if we took out, for example, Synechococcus, can we tell if that's really the driver of the ecosystem? I think with that technique is really just a correlation metric. So I don't think we can really get at drivers with this metric. Of course, if we were able to make samples where we dropped out Synechococcus, I think that would really start getting at these questions and manipulating the community. But we haven't really done that yet.
Mm-hmm. Thank you.
To disentangle the circadian part of the response versus just the acute light response, can you do things like just shine light on a part of the mat continuously, or maybe shade it, or how are you thinking about doing that?
You're right. So disentangling light response for circadian. So I think in situ, there are some really nice studies back in the 80s and 90s where people did actually do some of this mat shading in situ. And now that we have more in-depth omics tools, I think it would be really great to go back to the mat. We are planning to try to do this more with some of our isolates we have in culture, where we can really more precisely control the day-night cycles in the lab. And we're hoping that we're going to get some mutants in our Synechococcus species in their circadian clock that we could start to disentangle that.
I think we have time for one more question.
So I was wondering if you are considering looking at mats somewhere else. So for example, in Iceland, it's been known for a long time, Castenholz found out that they are missing some of the phototropes that are present in Yellowstone. So if you would compare mats from Yellowstone with mats from Iceland, similar conditions, you might be able maybe to figure out the role of individual microbes. I don't know.
Yeah, no, that's a great point that kind of the different hot spring systems do have some things in common, but some of the individual players are quite different between the mats. It's not something our lab has planned to do, but I think there's a lot of work in the field comparing the different mats.
Okay, great. Let's thank Amanda.